All right, biology students. Today we're going to just kind of continue with our notes on genetics. So today I just need you to kind of pay attention to this video and just look through a couple of different problems I'm going to work out for you, which will help you with the homework for today. So as you guys will remember, hopefully from last week, we just did a whole bunch of genetics problems, involving just your regular. I'm trying to find a place to move myself here. Here we go. Just like regular dominance and recessiveness. So, for example, if I say cross a heterozygous green plant with another heterozygous green plant, this was a problem that we actually worked out previously. And you would just set up your Punnett square, right, and then just fill everything in, and then you can come up with your genotypic ratios and your phenotypic ratios. So, I mean, we've done, you know, dozens and dozens of these problems up to this point. So, I kind of think we're probably ready to move on to maybe some different types of Punnett squares. So, the first one that we're going to look at today is something called incomplete dominance. Incomplete dominance, a really good example of this is there's a type of flower called a snapdragon, and the color of the flowers is determined by something called incomplete dominance. So we're going to kind of go through this chart together, then we'll work out a problem, and you'll see that it's actually pretty easy. But just like every trait that we've talked about to this point, there's one gene that controls it. In this case, we're going to call it the R gene. We can call it any letter we want to, but I'm just going to pick R, whatever. Now, like again, all the traits that we've looked at, there's been like a dominant and there's been a recessive allele, right? Well, the thing about incomplete dominance is neither allele dominates the other one. So there isn't a recessive allele because there's not one that dominates the other one, right? So instead of writing a capital letter and a lowercase letter, we're going to use this little symbol here called the prime symbol. Kind of looks like an apostrophe. We're going to use that to help us distinguish between the two different alleles for this trait, because in snapdragons there's two different colored flowers. There's red flowers and there's white flowers. Red, we're going to use the R prime symbol, whereas white, we're just going to use the R symbol. All right, so that's a little bit confusing. It won't be after we do this. You'll see. So, we're going to have, again, different genotypes because each parent can pass one of these two alleles to their offspring. So, one example would be R prime, R prime. That means both parents pass that R prime allele. Then we have RR. So, both parents pass the R allele. Then we have R prime R, where one parent passed the R prime, the other parent passed the R. So now here's where this gets a little bit different. Okay, so if it's R prime, you would imagine it's red because these are both red alleles. So the flower color would be red. If a plant has two white alleles, it's going to be white. But if a plant has one of each, now we've been thinking up to this point that this would be a red plant, right? But this is incomplete dominance. What happens during incomplete dominance is neither one of the colors show up. Instead, they kind of blend together. So if you have a red parent, R prime, R prime, and you have a white parent, RR, all of these offspring are going to be pink. They're all going to be R prime, R prime. Okay. So that's what kind of makes this one a little bit different, because the heterozygous condition, where you have one of each, normally, again, it would be the dominant phenotype, but in this case, the colors kind of blend together, so you get pink. Let's go ahead and do a Punnett square for this. So let's say that we want to cross two pinks, a pink snapdragon with another pink snapdragon. So here's your two genotypes, right? So we're going to go ahead and fill in our Punnett. R prime R on one side, R prime R on the other side. So if you look at the boxes, right, this offspring is going to be red. These two are going to be pink because R prime R, and then this one is going to be white. Okay. So again, just like always, you can do your genotypic ratios and your phenotypic ratio. So genotypically, 1/4 or R prime, 1/2 or R prime R, and 1/4 are RR. And then our phenotypic ratio, 1/4 red, 1/2 pink, 1/4 white. Okay, so incomplete dominance, that's when both colors kind of blend together. If you kind of remember that, it'll be a little bit easier for you. All right, now here's another example called codominance. Now, codominance is a little bit different. A good example of this is coat color in cattle. It's also found in horses and some other animals too. But the coat color in cattle is determined by a gene, just like all the other traits. So we're going to use the C gene. Now, the thing about this one is codominance means that they're equally dominant. That means they both dominate. They don't blend together. They kind of both show up, and I'll show you this in a second. But here's our two alleles. All right, we're going to use CR, kind of a superscripted R, um, to denote red or red cattle or red cows or kind of like an auburn colored cow. Then CW is white. Okay, so we have two different versions of this allele. So of course we have our three genotypes, right? CR, CW, and CR. So now what we get with this is we get again, kind of what we would expect with CR. We would expect red. CW, we would expect white. Now, CW is something called roan. Now, we have to be a little bit careful with this because roan does not mean that the colors blend together. Roan means that they both show up. So, for example, this cow is brown, or, well, it's supposed to be kind of reddish color, but it'd be kind of red and white. So instead of like, it'd be like, if if this cow was red and this cow was white, if this was incomplete dominance, then this cow would be pink. All right. But in this case, both colors show up. They don't blend together. All right. There's a little bit of a difference. So for example, like if I took like a razor and I shaved some of the hairs off of this cow and I looked at the individual hairs in my hand, I would see brown hairs and white hairs. Are they both show up? Okay. They don't like blend together to give like a tan color or something. All right, so that's something I kind of want us to understand. Well, there's a little bit of a typo here. This should be codominance. Let me change that real quick. Codominance. There we go. I always catch my little mistakes when I do these things. All right, so let's do a problem with codominance. So let's say we're going to cross, I don't know, in this case, a roan bull with a roan cow. All right, so they're both roan, right? So there's another typo. Fix my little superscript here. There we go. That looks better. I'm kind of a perfectionist when it comes to my Punnett squares. All right, so here's our two parents, right? They're both roan, right? So CW for both. So again, we put those two parents, one on the left, one on the right, and we just fill it in, right? So this offspring would be a red cow or a red calf. These two would be roan. Now notice how I colored this. Now it's red and white because they both show up, okay? And then this one would be white, right? So when we do our ratios, 1/4 would be CR, 1/2 CW, 1/4 CW, and then 1/4 red, 1/2 roan, 1/4 white. All right, so this is codominance. It's a little bit of a difference from incomplete, but I mean, they're Punnett squares. They're not that difficult to do. As long as you know how to write the parents and fill in the Punnett, you can figure out any of these um, ratios. All right, now the last one that we're going to do today is something called multiple alleles. All right, really good example of this is blood typing. I'm not sure if you guys know what blood type you are, or even if you know how many different blood types there are out there. But we're going to talk about this today. Now, blood typing, again, is caused by one gene. Okay, we're going to call it the I gene. Now, again, like every trait, there's different forms of the gene. Now, notice in this case, we have 3 different versions. Everything we've done up to this point has only had 2. I don't care if it's regular dominance or codominance or incomplete, there's always 2. But in this case, there's 3. So that's why we call this one multiple alleles. So now I want you to think about all the different combinations of genotypes you can have. Remember, every parent passes one of these to their offspring. That means that we all have 2, right? We got one from our mom, one from our dad. So one is what we call IAIA. This is IAIO, IBIB, IBIO, IAIB, and IOIO. Okay, so there's 6 different genotypes that are possible. All right, now the phenotype is what blood type you have. All right, so for example, if you have IAIA, you have type A blood. Okay, no matter what, you're type A blood. If you're IAIO, you also have type A blood. And the reason why is because O is recessive. So if you have AO, it's going to be type A. IBIB would be type B blood. IBIO would be type B blood because, again, O is recessive. So if it's BO, it's going to be type B blood automatically. Now, here's where it gets a little bit crazy. IAIB. A and B are codominant. So if they're together, they have their own phenotype. This is called AB blood. And if it's IOIO, of course, it's type O blood. All right, so I want us to understand the different genotypes. There are 6 of them, but from those 6 genotypes, there's only 4 phenotypes. So I want us to know the different combinations and what phenotypes those would correspond to. All right, so that shouldn't be that difficult though. All right, let's do a Punnett square. All right, blood typing. Cross a man or a male heterozygous for type A and a woman with type AB. So here's your parents. Now, heterozygous means that you're AO. Okay, if it's homozygous, you'd be AA. And then the other parent is AB. So, of course, we just pop those in there. And then we go ahead and just fill it all in, just like we always would. Okay. So if you look at the offspring here, right, this would be a baby who's type A. This one here would also be a baby who's type A, but this one is heterozygous and this one's homozygous. This would be an AB baby, type AB, and this would be a BO. So if we're going to do our genotypic ratio, right, 1/4 IAIA, 1/4 IAIO, 1/4 IAIB, and 1/4 IBIO. For phenotypes, you're telling me what blood type they are. So, it would be 1/2 type A blood. There's 2 out of these 4 type A blood. 1/4 would be type AB, and 1/4 would be type B. Okay. All right. So there's 3 different patterns of inheritance that we talked about today. We talked about incomplete dominance, codominance, and also blood typing, which is multiple alleles. I have another typo here. I should probably change this. Anyway, so I wanted you guys just to kind of be aware of those different types of inheritance. So when you do the homework today, you can use this particular PowerPoint if you want to kind of help you out. All right, guys, have a good day, and I will see you soon.